All right, so yesterday we got through all the parts of the cell and what they do, and we just kind of started diffusion. So I'm going to start diffusion over again. Okay, go over what diffusion is, why it's important. We'll move on to osmosis and active transport. Those are the big three, okay, or what we call the big three methods of cellular transport that we are most concerned with. Okay, we're looking at diffusion, we're looking at osmosis, and we're looking at active transport. They are all different. Okay, I mean, a couple of them have something in common, but they're quite different in terms of the entire process. Now, in terms of diffusion, okay, diffusion is what a cell uses to transport material from one place to another inside the cell. Okay, that is, it is responsible for intracellular transport. Okay, essentially, all transport within the cell is accomplished through diffusion. Okay, there's several reasons for that, but the most important reason is that diffusion gets the job done and costs the cell nothing. Okay, no energy is required for diffusion to occur. Okay? Diffusion is caused by the natural movement of objects, of molecules, things like that. Okay, they generally want to naturally move from an area where they are crowded and concentrated to an area where they are less crowded and concentrated, right? So their movement, their natural movement and kinetic energy results in them kind of bouncing off of each other and naturally spreading out, right? The further they have to travel, the more spread out they are going to become. Is that going to be a problem for something the size of a cell? Not usually, because cells are pretty what? They're pretty small. Okay, that does, however, set an upper limit on the size of a cell. If a cell gets too big, diffusion doesn't work very well, because by the time materials have transported from one side of the cell to the other, they've traveled so far that they're very spread out, and not all of the material is getting where it needs to go. Right? Generally, things will get where they need to go because of diffusion, because it's going from high concentration to low. And where they're needed would be low concentration. Where they're produced, they would be in high concentration. So it generally works provided the cell isn't too big and the material doesn't have to diffuse very far. Okay, everybody with me there? Okay, so in that way, diffusion works really well for what the cell needed to do. There are drawbacks to diffusion. It is not specifically targeted. Okay, that means that instead of being like, all right, I'm sending this package from this address to that address and it's going to get there, it's more like the shotgun approach. Okay, I'm going to shoot this shotgun and some of it will get to where I need it to go. Okay, that's how diffusion works. It kind of spreads out and gets everywhere. All right, so it's not specifically targeted to one location. It's also quite slow. Right, so it's like, if you choose the free shipping off of eBay, right? Like it takes forever. It's like 10 weeks before it finally arrives at your house, right? But again, does anything have to travel very far from one side of a cell to another? No. So even though it's slow, we're talking about incredibly small distances. Even if you're traveling slow, you can still get from one spot to another if they're only a few micrometers apart fairly quickly. But again, that contributes to the upper size that a cell can reach. Okay? It is diffusion that really determines the maximum size of a cell. Okay? If it gets too big, this is too slow and not specifically targeted enough to get materials where they need to go fast. Okay? All right, so simple rule of diffusion is this. In the absence of any other forces, and that's important, there's no like pump or current or something like that, okay? A substance will diffuse from where it is more concentrated to where it is less concentrated. We call that down the concentration gradient. Okay, essentially it's downhill. Anything that's free is only going to go downhill. Right? It's not going to go uphill. All right, it is passive, so it requires no energy from the cell. All right, everybody okay with that? All right, so what can diffusion transport? Everything. Yeah, basically anything can diffuse. Right? It's a natural tendency of all molecules to go from where they're highly concentrated to where they're less concentrated. So diffusion can transport anything. That also makes it really useful. Okay? There doesn't have to be a specific method to transport this material and another method for this material. Everything can move by diffusion. All right? So diffusion's inside the cell. It can transport anything. It's good over small distances. It's not very fast. It's not targeted. Okay? Those are kind of the things we need to remember about diffusion. All right. Now, the second of the big three cellular transport methods is called osmosis. How many people have heard of osmosis before? Okay, probably in the context of reverse osmosis, right? Okay, that's used to make drinking water. We're actually going to talk a little bit about that tomorrow. No, maybe tomorrow, we'll see. But it's the passive transport of water only. That's the thing about osmosis. Okay, it will only move water, nothing else. Okay, so that's kind of a limiting factor for osmosis. Okay, it's passive, but it's only water, so it's free. The drawback of, again, being passive is that the cell has no control or virtually no control over this process. It's just going to occur because it's a natural process that is passive. Okay? That can be good or bad. Good in that it happens and the cell doesn't need to worry about it. Bad if things get out of hand. Okay? And by out of hand, I mean if we're moving water, we're trying to achieve some kind of equilibrium. In fact, the other cellular transport process, diffusion, is also trying to achieve an equilibrium. All the passive processes try to achieve equilibrium. It's really concentrated here, but as they spread out, they become equally concentrated everywhere. Right? What osmosis is trying to do is balance the amount, or the concentration of solutes, specifically salts, on two sides of the same membrane. Okay, so if I have the inside of a cell, there's always some salts dissolved in the cytoplasm and stuff on the inside of the cell. Outside of the cell, that can vary depending on environmental conditions. Everybody with me so far? What osmosis does is sends water either in or out of the cell across the membrane to make sure the concentrations are equal. So if I'm in a lifeboat in the middle of the ocean and I drink a bunch of salt water, how is that going to affect me? Exactly. I'm going to get dehydrated and I'm going to feel thirstier because if I consume a bunch of fluid that has lots of salt in it, the concentration of salt in my blood and in my tissues is going to go up. Okay? My cells, which have no control over this process, are going to start to lose water because water is going to move across their cell membranes into the saltier stuff, okay, into either the blood, mostly the blood, okay, or the other types of tissue. So water is going to leave the cells and go into usually the blood. Now, if more water starts leaving the cells and going into the blood, okay, my body will detect that. Specifically, my kidneys will detect that. And they'll notice that my level of, or volume of like plasma in my blood, okay, the amount of fluid in there is going up. And that's not always a good thing. So what will my kidneys start to do? No, they won't shut down. They'll do their job. They'll start taking water out of my blood. That's what they're supposed to do. Okay? They'll detect that there's some salt, but there's an excess of water. Okay? Because water is being drawn out of my cells. So my kidneys will pull the water out of my blood, and then what happens to it? I lose it. You pee it out. It's all gone. Okay? That's not going to help my situation, is it? I just drank, let's say, a liter of salt water. I probably lost 1.5 liters to get rid of it. Okay, to get rid of all that salt that I took in. So I'm actually now 0.5 liter of water in the hole, okay, as a result of drinking salt water. Okay, so it's just osmosis. Osmosis is going to draw that water out of your cells in order to make sure the concentration of salt on each side is the same. Okay, now I've lost all that water. I'm going to feel thirsty. So I'm going to drink some more salt water. Am I making the situation worse and worse and worse? Yeah, I just keep putting more and more salt in and I have less and less water to get rid of it with. Okay, eventually I just get really dehydrated and I'll turn into a raisin. Okay, I'll find my desiccated carcass in that lifeboat sometime. Right, so I want to avoid drinking salt water for 2 reasons. First off, it's going to dehydrate me. Secondly, the ocean is the world's toilet. Everything flows to the ocean. Don't drink ocean water. Okay, yeah. Gross. Right? So you're actually, even grosser, better off to drink your own urine for the first couple of days that you're stranded in a lifeboat. Till your urine gets too concentrated, then it's not good. Okay? But for the first couple of days, there's actually less salt in your own urine than there is in the salt water. Might buy you an extra day.
Are cells only on the inside of your body?
No, they're everywhere.
So what happens to a cell when it gets cut on your hand or something?
Oh, cells rupture. Yeah.
So
They die. Yeah. Okay, so looking at the process of osmosis, like we said, big thing here is the passive transport of water. And it's not only of water, but it's also across a membrane. Okay, this process doesn't happen within the cell. Water doesn't need to be transported within the cell. The cytoplasm is full of water. Okay, so this is moving water across the membrane, and the reason to do that is to balance solutes. Okay, that's what osmosis does. Okay, and remember, this is not a process that your cells have any control over. It's just going to happen. So if I took a cell and put it in what are called hypertonic conditions, that is, I put it in really concentrated solution of salt, of some kind of salt, it's going to shrivel up, just like a slug would if you poured salt on the slug. Okay, it'll shrivel up because all the water is going to come out of it to try and balance the salt. Okay, so it'll just shrivel up. The opposite would happen if I took a cell and put it in distilled water, because there's no salts in distilled water, but are there some inside the cell? Yeah. So now what happens is water moves into the cell instead of out. But since there's no salt outside of the cell, can a balance ever be achieved? No. You can't balance something if you got nothing on the other side. So water just keeps going into the cell until the cell pops. Okay? You can actually rupture a cell if you do that. Right? We don't have to worry about that because we're quite a bit more complex than just a single cell. Okay. All right. So this is a lab that you'll actually do in like Bio 20. Okay, where you set up an apparatus like this. You have a U-shaped tube, and there's a piece of like sausage casing that goes across in the middle. Okay, sausage casing is made out of intestine, so it's cells. It's essentially just cell membrane. All right. On one side of that sausage casing, you'll have highly concentrated salt water, and on the other side, very low concentration salt water. And what you'll see over the course of even like one class period is the water level on the low concentration side will go down, and the water level on the other side will go up, because water is moving across the membrane by osmosis in order to make the concentration on both sides the same. Eventually, that movement of water will stop, and when it does, the concentration of salts on both sides of the membrane will be the same. Okay, everybody kind of follow there? Reverse osmosis works in reverse, hence the name. Okay, it takes water and pumps it, forces it across a selectively permeable membrane like this, and leaves all the salt on one side, only getting pure water on the other, right? But that's not a passive process, is it? Okay? It's being pushed across or forced across with a pump, so it's not a passive process. It's also not a natural process. It's entirely artificial. Okay, there are no cells that go, oh, I'm just going to clean some water and do some reverse osmosis right now. Okay, it's an artificial process. It's an artificial membrane. Okay? It's entirely different. You can try to distill it. The one that I've seen that worked best was you dig a hole in the beach. You have to be on an island here, right? Dig a hole in the beach, and you fill the hole like with the water, right? And you cover it up with like something black garbage bag, something like that. And as the water evaporates, you can like put a rock in the middle of it so that the evaporated and condensed water will catch on the garbage, and then you put like a cup in the middle, so the purified distilled water essentially that's condensed on there collects in this cup. It's a slow process. You can do that with salt water or urine or anything. Okay, it's just distillation. Right? To use reverse osmosis, you'd have to have a pump and a membrane, which, unless you were really well equipped, you probably wouldn't have. All right, does everyone understand what osmosis is? Okay, the key points for osmosis are that it only transports water. Okay, so if I give you a question on a unit exam that says calcium chloride needs to be removed from a cell, is it going to be this process? No, this process only moves water. Okay, all right. And the last of the big three is active transport. Active transport is much different than the other two because it's not passive, it's active. That means it requires energy from the cell. Okay? Your cell can actually actively pump certain materials through the membrane. Okay? There are little proteins. Remember I showed you what the cell membrane looked like yesterday? It was like Jell-O salad. It had lots of stuff kind of embedded in it. One of the things that's embedded in the membrane are called carrier or transport proteins. Okay? And these are like gates. The cell can use energy to change the shape of the gate. It can either be closed or open. All right? So if a cell needs to pump a material into the cell quickly, it will use this process. It'll use active transport. Okay? And it can pump material fairly quickly using this process, but it's against the gradient. Okay, so where the other two were kind of letting everything go downhill naturally, this is moving stuff the other way. It's not going from a high concentration to a low concentration. It's going from a low concentration to a high concentration. It's backwards. That's why it requires energy. Okay, so what happens is the energy is used to essentially change the shape of these carrier or transport proteins. So here's kind of their natural state here on the left, and there's these ions, whatever they happen to be. They get into the gate, and then energy is used to change the shape of the protein, and it closes off to the outside, opens up to the inside, and the material is then actively pumped through the membrane and into the cell. Okay? Everybody follow there? Right, so active transport uses energy. So it's the pumping of solutes against the gradient. Right? It's active. Okay, so it's going to use energy. Okay, it's actively pumping the substance across the membrane. That's the other thing. This isn't intracellular transport. This is getting stuff into the cell from outside, or sometimes from inside the cell to outside. Okay? Muscle cells and nerve cells use this process a lot. Okay? Every time a nerve cell fires, okay, sodium and potassium ions switch places across the membrane as the electrical or the impulse is sent through. Okay? Before it can send another impulse, those have to be pumped back the other way. Okay? So nerve cells and muscle cells use active transport to pump the sodium and potassium ions back and forth in order to be able to send impulses and contract in response to those impulses. Okay? So active transport is used quite a bit with muscles and nerves. Okay, so don't forget this carrier protein part here. Okay, that's the thing that's actually responsible for the pumping of the ions across. Okay, questions on active transport? Okay, so the big three: diffusion, osmosis, active transport. Okay, those are the big three. What we're going to look at now are some ways that the cell can move things in or out of the cell involving changes in the shape and structure of the membrane. Okay, this is only changing the shape and structure of a carrier protein, not the entire membrane. Right? When you guys were looking at the amoeba there the other day, okay, the amoeba consumes food by a process called phagocytosis. It means to engulf something and physically put it inside of a vesicle. Right? So how many people have walk their dog? Okay, and you have to pick up after it, right, when you walk it. Okay, so you put the little plastic garbage bag thing over your hand, okay, and you pick up the leavings, and then you turn the bag inside out. Right? That's exactly how a cell takes materials into it. Okay, it engulfs the material and then turns the membrane inside out. Okay, now a vesicle can move into the cell. It's a food vacuole now, right? It'll merge with a peroxisome and it'll become a lysosome and digestion will occur. All right, everyone kind of follow me there? That's the process of phagocytosis. So it's taking stuff into the cell, okay? It's endocytosis, but it involves changing the shape of the membrane, right? So it looks, sorry, exocytosis was first. Exocytosis is the exact opposite of what I just described. So a vesicle comes from the Golgi apparatus, okay? So it's enclosed. It's basically just an envelope that's made out of membrane. Okay? It goes to the membrane, and when it makes contact with the membrane, it just merges with it, and whatever was inside the vesicle just pops out and into the fluid between the cells. Okay? Right, so that's how all exocytosis works. The process is that's how everything gets out of the cell from the Golgi, right? There are three different ways things get in through this endocytosis, okay? We just talked about phagocytosis, okay? So that's right here. This is an amoeba, and it's engulfing that food particle, so it puts it into a vacuole, okay, like this. So it engulfs first and then has it inside. Okay. Phagocytosis is a very common process for cells to use to engulf large food particles. It's also what your white blood cells use to engulf bacteria and other things to kill them when you're fighting off a cold or an infection. Okay. They engulf that stuff and they actually use it as food. Okay. The other two kinds of endocytosis are pinocytosis. They're all mechanically the same. The membrane just engulfs something. Pinocytosis is just the engulfing of fluid. It's like this is cell eating, this is cell drinking, right? A cell will do this if it has become very dehydrated and suddenly a lot of fluid is available. Okay? Rather than wait for osmosis to slowly, passively move things in, it'll just start gulping. Okay? Gulping a whole bunch of fluid and letting that fluid get into the cell. Right? It would be just exactly what you would do. If you've been out in the sun all day and you hadn't had a drop of water and somebody hands you a nice cold glass of water, you just chug a lug. Okay, well, this is what the cell is doing here. Okay, the third way uses the receptors. I said that on the surface of the membrane, there's all these little antennas that accept the signals and stuff from the body. Okay, if a signal is received by a receptor, then the cell membrane will engulf the signal. Because usually those signals are large proteins and they're too big to just get through the membrane, so they actually have to be physically engulfed. Right? Mechanically, these things are all the same. Something gets engulfed and taken into the cell. What's different is what's getting engulfed and taken in. All right, so like we said, phagocytosis is a cell engulfing a food particle by wrapping what's called a pseudopod, which is a false foot. I don't know why it's called that, but that's what they're called. Okay, around it and packaging it within a membrane enclosed sac large enough to be called a vacuole. So this is one of your immune cells doing that to a bacterium. Okay, in pinocytosis, okay, it's same process mechanically, but we're just engulfing fluid now. Okay, so we've got all this interstitial fluid, that's fluid outside of the cell or between the cells. It just gets engulfed, and we've got a vesicle coming in that's just full of the fluid from outside. Okay, this is what it looks like under an electron microscope here on the right. Okay, so we can see this is the cell. So this is the cell membrane here on both sides, and you can see the vesicles being formed. They just kind of engulf in and then they just pinch off, right? And then they form a vesicle. So this one here is just about to kind of pinch together and form a vesicle. This one here is probably just forming. Okay, so it's just filling up now. Okay, that makes sense? Okay. And then lastly, we've got receptor mediated endocytosis. So this doesn't happen unless one of the receptors on the membrane says, oh, we just received a signal about blah, 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 engulf it, bring it in. Okay, so that's essentially what they're doing. So you got these receptors that are on the outside. When the chemical signal is received, the cell membrane engulfs it and takes it in and sends it to wherever it needs to go within the cell. Okay, this is what that looks like under an electron microscope. So this kind of fuzzy stuff here is the outside of the membrane. This is one of the receptor proteins right here. Okay, they're all kind of shaped like a cup almost. And I'm just going to erase my drawing so you can see it behind the ink there. And it's kind of a black, kind of a dark black thing there, and you can actually see the molecule that it's grabbing is kind of touching it there, a big long hormone or something like that. Okay, once those receptors, and there's a whole bunch of them along here, have received those signals, then you see that the membrane very quickly changes shape to form that vesicle. And you can actually see those receptors quite a bit better, including the molecules they're holding. Okay, in this picture here, there's a whole bunch of them there, and they just get engulfed and taken into the cell. Mhm. No, they bring them in and kill them. They essentially eat them. Yeah, they just, the lysosome comes over and, yeah, just digest them. Yeah, I mean, it's essentially just recycling it, right? Like, I mean, a lot of the stuff will just be destroyed, but a lot of it could be used as food and whatever, and then, nope, no, it would. Bacteria don't necessarily harm a cell. Bacteria make us sick because as they carry out their life processes, they release toxic chemicals. Okay, so if I, go and eat at Arby's and I get salmonella, okay, the salmonella bacteria release something that makes me horribly nauseous, have all kinds of intestinal distress and whatever else. Okay, that's what the effect of that bacteria is. Tetanus bacteria, they release a toxin that doesn't allow my muscles to relax. Okay, so as long as I can go out and have my immune system find those bacteria and kill them, I'll be okay. With a virus makes you sick by killing your cells. Right? It gets inside, makes your cells make more viruses, and then when they get full, they pop and die. Okay, so it's harder to fight that for your immune system because it has to go about killing your own cells. It can't find individual viruses. It kills infected cells instead. Yeah. Okay. Everybody follow there. So the big difference here with receptor-mediated endocytosis is that it is specific. It happens when a signal is received as opposed to I'm going to gulp in some water or there's a piece of food over there, okay, or whatever. All right, I want you guys to answer those questions. I'm going to give you a few minutes to answer those questions. Just do your best. The answers aren't necessarily word for word in the notes here. I want you to think about them a little bit because that's what biology is about. I memorize some things and then I figure out ways to apply them. So here's kind of the first exercise in that. All right, so let's have a look at these questions here. So question number 1, color blindness is caused by a person's cells being unable to produce certain proteins, not all proteins. Okay, so where in the cell does the problem lie? What's not right in here? How many people were thinking it might have been the ribosomes? Okay, if it was the ribosomes, though, they wouldn't be able to produce any proteins, right? This is only certain proteins, okay? If someone's colorblind, why are they colorblind? It's genetic. Color blindness is genetic, okay? The problem lies in your DNA. Somewhere in the DNA, there's a mistake, right? And so even when the call goes out for this protein to be produced, the ribosomes don't produce the correct thing because they're not getting the right information from the DNA, right? So that leads to a person being colorblind. Not the nucleus, it's actually the DNA. The problem is in the DNA, okay, which is of course in the nucleus, but the problem is in the DNA. Okay, Mr. Van Erve, who's just 2 doors down here, teaches biology, is completely colorblind. Okay? We once actually played a joke on him. He came in one day and he was wearing like a certain pattern of shirt, and there was like two of us who were also wearing the same pattern, but all 3 of us were wearing different colors. We're like, hey, Jerry, look, we're all wearing exactly the same shirt. And he's like, oh, that's great, and everyone's going around, hey, we're all wearing the same shirt. What are the chances of that one? Everyone's looking at him like, what? Jerry, like that shirt's purple, that shirt's red, that shirt's green. And he's like, so he wants to get those glasses that apparently fix color blindness. Okay, yeah, with that. Yeah, apparently they work. I haven't seen them, and of course I wouldn't know because I'm not colorblind. But okay, for Mr. Van Erve, his genes are that he has no ability to see any colors at all. He's like the most extreme end of color blindness. Most color blindness is like this color and that color don't get quite aren't quite right when you look at them. But I mean, you're never going to know because you've never known any different if you're colorblind. Most people don't find out they're colorblind until they take their driver's test. Okay, and when you take your driver's test, they make you look at these little patterns. Okay, and if you're not colorblind, there's dots in them, and the dots are different colors. And if you're not colorblind, you can see the numbers that the dots make up. If you're colorblind, it's like I just see a bunch of dots. Okay, Mr. Van Erve, when he looks at all of those, just sees dots. He never sees any numbers in the dots. Okay, now is it a big deal? Like, are you going to not get your driver's license if you're colorblind? Well, no, you'll still get your driver's license. That's why red is always where on the stoplights? Either on the very top or on the far left, always. Okay, green is always on the far right or on the bottom. Okay, the only problem would be is if you got one of those like the advanced green, right? The advanced green arrow turns yellow when it's about to change over, but it also goes from flashing to solid. See, there's all these things that have been put into place to account for the fact that many people are actually colorblind. Okay, Mr. Van Erve comes up, he knows, oh, that light's about to change to red. Red in his mind. Okay, whatever red is to Mr. Van Erve. Okay, because he knows it stopped flashing. It's still lit, but it's not flashing. Right? So there's all these things that are put into place to help account for people who are colorblind. Okay. In Canada, like we have all the different colored money, right? Even Mr. Van Erve, it all looks the same to him, except that it's got different numbers or prime ministers or whatever on it. Okay, he's great in the States where all the money's the same color. He doesn't. Like if we say, Jerry, that's purple, he's like, okay. Not really. I mean, he'll say, that's purple, and we're like, no. Yeah. No. Yeah, like, no, you can tell, like, if he gets out of the house before his wife sees him in the morning, okay, you can tell, because he'll come in with, like, 2 different color socks, or, like, a shirt and pants that don't match, and it's not his fault, he can't tell that they don't match, because he can't see the colors. Okay? What's that? Or shades of gray. Yeah, I mean, it's hard for him to explain, because he doesn't know any different, right? He's never really seen those. Well, he can tell me that his car is gray, and he's right. I think that's part of the reason he bought a gray car. He likes to be right. But yeah, so color blindness is genetic, and it's far more common in men than it is in women, because the gene for color blindness is on the X chromosome. If you're a guy, you only get one X and one Y. All right. So if you have the color blindness gene, you're colorblind. If you're a girl, you have 2 X chromosomes. So you could have one X chromosome that has the colorblind gene. As long as the other one doesn't have it, you won't be colorblind. So it is far more likely for men to be colorblind than women. Okay? But it's genetic. Okay, why is it important for organelles to have their own membranes? Well, if we're talking about the nucleus, it's an extra layer of protection. Yes, it wasn't really what I was thinking of, though. Okay, what did we say was important about compartmentalization? Okay, this is the question that's asking about compartmentalization. Right, having the fridge and the freezer and the area where you live, they all need separate environments. Okay, having their own membranes means they can create their own ideal environment in which whatever job they have to do gets done best. Right? So for the lysosome, a slightly acidic environment with digestive enzymes. For mitochondria, it's a warm environment where sugar is broken down and things like that. So having their own membranes allows them to make their own little environments where their process occurs. Okay, so essentially it's compartmentalization is the answer for number 2. Okay, number 3, if the lysosome's membrane ruptures, what happens to the cell? It dies, because what's inside the lysosome? Digestive enzymes whose job it is to digest cellular material. So if the lysosome ruptures, the cell will eat itself to death. Okay, yeah, probably not the way I would want to go. All right, lastly, what problems might a cell have if all the ER was removed? Don't say the cell will die. Yes, why? What is it not going to be able to do? Okay, it's not going to be able to detoxify poisons. It's not going to be able to transport proteins or lipids. What else? Well, that's a different thing. No, rough ER transports protein. Yeah.
It won't be able to repair.
Can't repair membranes, that's what you're going to say. Okay, and wouldn't be able to break down complex carbohydrates. I would say probably the first and largest problem the cell would have that would lead to its demise would be the membranes. Okay, that's the primary job of the smooth ER, is to make lipids so membranes are constantly being repaired. As soon as you stop repairing membrane, okay, it's like saying, oh, well, I've got this big wall designed to keep stuff out, but I'm not replacing the holes in it. Okay, I'm not fixing any of the holes. Well, once it gets a hole in it, it really ceases to do its job. Okay. Mhm. Yep, that's exactly how it does it. It ruptures the lysosomes. It's called cell lysis. Yeah. Okay, questions on any of those? All right, so this will hopefully give you some idea of the kind of some of the ways I might ask things on a unit exam. Like asking you what would happen if there was no ER in a cell is kind of another way of asking you what's the job of the ER. Okay, rather than phrasing it that way, right? You answer it a different way. Okay, so that's the kind of thing that we're going to be looking at. Tomorrow, we're going to be working on an in-class activity. That means you're going to need to get it done in class tomorrow. Right, so we'll have the Chromebooks and you'll be working on that. Okay, so you'll need to stay fairly focused and on task for that one. Okay, so come ready for that in the morning.